Thanks, Chloe, for the introduction. First of all, I'd just like to say thank you, everybody, for joining today's session. And let's just get this started. So today's topics will be as follows. So we'll go over a brief introduction on Illumina-based sequencing. I say brief because this topic can take several days to master. We'll then proceed to library structure and type, give you some details on our sample requirements and how we perform our library QCs, the way we demultiplex and sequence, and the variety of options that are available, and any other lingering topics that may arise. So let's go ahead and start out with the introduction to Illumina-based sequencing. Essentially, all that is needed to be done, and I say all as simplistically as I can, all that is needed to be done with your area of interest is to take a DNA insert and add adapters to it on either side, flanking. So on this end, we have our 5' end, so our P5 adapter, which will bind to the flow cell, has been added to our DNA insert. And likewise over here with our P7 adapter. So this allows us to do sequencing by synthesis on the machines themselves. So we go from taking our area of interest and adding adapters to it, to clonal amplification. So theoretically, the way this library is to be, so this is called a library, and theoretically the way this library is to be loaded onto one of two machines, if we are talking about HiSeq, then it is to be loaded on our CBOT, which is the cluster generation machine. Or if we're talking about Novogene, it is loaded directly on the sequencer itself. So the sample, the library that is generated, is theoretically placed on the lane. Within the lane, there are several tiles. And those tiles are where the clonal amplification occurs. So theoretically, one molecule should generate an entire cluster of identical molecules. And we see that here with this depiction. So these individual dots here are colonies that have generated from theoretically one molecule. And so that one molecule will have a unique indice set if it is to be demultiplexed. One thing to point out here is we can see that these aren't pretty, dots. They're pretty amorphous. They're all over the place. This is typical for NGS sequencing. The machine itself will decipher the calls based on signal strength. And that is how we go from library to cluster, and then eventually over here to our data recognition. All right, so we have constructed our library. We have potential, depending on which platform we are on, we have created clonal libraries. If this depiction here, this is the flow cell for a HiSeq lane, a HiSeq run, and there are 8 lanes within one flow cell of the HiSeq platform. On this lane, there is an oligo lawn. So this is where our libraries will covalently bind onto the flow cell. Now this gets back to right here. So we have bound, and in this case, in the HiSeq, we have bound and we have generated clusters. And so as we can see, a couple of depictions here are the fact that these colonies don't perform or make the best clustering, and that's okay, because once again, we have some overlap here, and theoretically this colony here and this colony here could be distinct. They could be different, or they could be the same genetic code, but that's okay. We'll be able to decipher this hopefully in the demultiplexing phase. So we've gone from loading the sample onto the flow cell. We've passed cluster generation. Now it's time to sequence. So the way the Illumina-based sequencing works is sequence by synthesis. So we have our oligos added here. We have incorporation of fluorescently labeled NTPs, right? So once we have that incorporation, a blocker or a terminator will be added such that another nucleotide cannot be added. So we can then only get one call at a time. If this wasn't in place, then this machine wouldn't be able to tell at what position or at what cycle this base is being determined. So it's important to know these distinctions here. All right, so we've gone from library generation to loading it onto the machine. And now we need to go understand the workflows that take place with Illumina-based sequencing. So here on our left-hand side, we can see that this approach is going to be single indexed. Now, why might you choose single indexing over dual indexing? A couple of reasons. If you don't need as much demultiplexing power, then single index is fine for you. If you have many, many samples and you are trying to cut your costs or potentially accommodate all your samples within one kit, then the ability to demultiplex should be greater here because you need to be able to separate the individual molecules themselves. So starting off with the single indexed sequencing approach, these both approaches are very similar. So essentially what happens here is we are going to bind our read1 primer. The read1 primer will extend at the initial, the first base of the insert, and we will get our read1 sequence. Then our I7 sequence will be run. That'll determine what indice is associated with what run. And then we are going to be reading the read2 primer. Attaching the read2 primer to get the read2 sequence. So once again, this is just single index demultiplexing. Over here, we have dual index demultiplexing. And this by far is the most common way that we see libraries presented to us. And this approach here is done on the HiSeq and on the Novogene platforms with the recently updated Illumina reagent kits. So here at Novogene, we will be using the V1.5 reagent kit. I'll go into further detail about that later on. But let's just continue to talk about the differences between these two approaches. So here we can see that the read1 primer anneals at the read1 site. It extends at the DNA insert. So the same thing is occurring in here as it is here. If we further continue to follow the same progress, so we go from the I7 identification, and then instead of going directly into the read2 primer sequence, we're going, the read2 index insert sequence, we are going to jump to the I5 index. So this allows greater demultiplexing power for your samples. So if you have a lot of them, you can differentiate them based on the differences between your I7 and your I5. Just want to point out the one thing here. This approach is new. So this step right here has been modified by Illumina to remove bridge amplification. So using this 1.5 workflow, we are able to skip that step, which will allow for greater accommodations later on. And I'll get into that shortly. So these are our two indexing workflows that Illumina has made. All right, so before we can get into the actual sequencing and the analysis and what is needed in our libraries and how to control for the data output, it helps to understand these workflows in greater detail. And so it is important to understand that Illumina is shifting from their reagent kit of the version 1 to 1.5. And so with that, first of all, it's a different set of reagents. And second of all, it allows us to increase the number of cycles within a given run. So let's get into that. And we'll also talk about a few more things. So here we are on the left-hand side, we have the old workflow kit. So this is the version 1. This is no longer being used by the vast majority of sequencing cores out there. So really the most important difference, in my opinion, right off the bat would be our Q scores. So just for some background, Q scores, they are Phred scores. And so Phred scores are a measure of the quality of the nucleobases themselves. So the higher the Phred score, the greater the probability of the quality you'll get in return. So we can see in the old workflow that the PE 150, the Q score is at greater than or equal to 75. But as we come over here with our new workflow, we have upped that significantly. So this is important, and it allows us to have greater confidence in our reads and the base calls themselves. So this gets back to this here is what I was alluding to earlier about the removal of the bridge amplification step. So here it is depicted in the workflow form. Here we have it written methodically as it progresses. So the forward strand, which is the workflow 1, goes as follows, right? The insert primer adds, we read the insert. The I7 primer adds, we read the I7. Apologies for that. And then in the first workflow, bridge amplification occurs. So this is where your actual library that's attached to the flow cell at this point will bend over and attach to another oligo. And then the second strand will be generated via bridge amplification. Now, the new workflow removes that step. So we go directly from reading the I7 primer to reading at this point, the second strand has been generated. So we do not go through bridge amplification. And we jump straight into reading the I5 primer. So here's the difference here in the two. Instead of the bridge amplification step that is done here, we do re2 synthesis. So we extend the opposite strand. And we can tell that it has changed directions because of the orientation of the P5 and P7s on the, this is the lane here. So allowing this to occur gives us ability to add extra cycles in the workflows, which can accommodate a vast majority of kits that are out on the market today. So as I just mentioned, so if we give an example of something that's commonly used today in the sequencing world, 10x, 8-tax-seq, very common library now, as we can see, the I5 index is split between 4, and then we have the I7 at 8. So the run cycles needed for this should be 150, 8, 16, and the 7 refers to lost indices. But essentially, we cannot perform this because the normal workflow only has the ability to accommodate 330 base pairs in total for a sequencing run. So we're missing out on some of the information, in this case, 1 base. We're missing out on some of the information here that we would like to capture in our DNA insert. So in this case, to make any modifications, it's not really that feasible if you're running 1 sample on the Novogene S4 flow cell on a lane. So the client or you would have to purchase an entire flow cell just to run this sample type. So this is a problem that Illumina noticed, and they have since helped to accommodate. Another option is to run it on the SP flow cell. That does have its benefits and drawbacks, but that should be evaluated based on the library type that is given. So this is the example of the 10x, 8-tax-seq library construct that uses the version 1.5 Illumina workflow. And so we can see that in this workflow, we have a total of 338 base pairs of reading. So that's 338 cycles that can be run on the machine itself. This allows for us to change in any way, really, theoretically, we can change the proportion of indice lengths. And this one caveat is that the indice lengths here, this is specific to S4, to the Nova-seq. The high-seq has its own limitations, and we'll get into that in a bit. So here we can accommodate greater cycles, which will allow for even greater demultiplexing power. If you can extend your indice length, you can create more diversity and more potential for individuality of your inserts. So in this case, with this new workflow, this can be done at a much, much cheaper cost on just the lane itself. And we also have some other pipelines, such as partial lane sequencing on our S4 platform that can be purchased by gigabytes. And I'll get into that in a bit. But so we are now able, with the new workflow, we are now able to accomplish sequencing greater indice lengths. All right, so we've gone over the general workflow. We now have a good idea of what the library constructs are, how they bind to the flow cells, how their clusters are formed, and a rough idea of how the workflow is to proceed. So now let's get into library structures and library types, the most commonly used ones, and how they affect our ability to get data. So at the top, right here, we have the generalized library structure. So let's just start from the 5' end. So the orientation that your library should read is 5' to 3', or P5 to P7. These here, the oligos, are what actually bind to the flow cell and allow for amplification to occur, and as well sequence by synthesis. In the workflow one, we would have seen a bridge amplification. So the P5 would have been bound and the P7 would reach over to another oligo on the flow cell and extend. But in this case, we are just producing the read to strand and going a different approach. So we have the oligos here. We have our indices, right? So index 1 is required for all demultiplexing needs. If you were to purchase a full lane, you don't have to demultiplex because you get the entire data output for that lane. But if you are to give us multiple samples and you want the ability for us to separate them, we have to have at least the I7 to get things done. So we have our two indices, and then it's right next to the read one primer sites. And then over here on the other side, so this would be on the reverse strand, the read two primer site. So the primers will attach right here, and the extension will be read at the first base of the DNA insert, and will extend in most sequencing approaches that we offer in Novogene would be paired end 150. So it would extend 150 base pairs in this direction, and on the reverse strand, it would extend 150 base pairs in this direction. Ideally, we would design our insert such that it is not greater than around 400 base pairs. We don't want the reads to overlap each other because then the data isn't as useful. So we've gone over the oligos, which are the P5, right? And so the oligos have to match exactly as what is Illumina available, right? So everything we do is run on Illumina chips. If the oligos do not match that, then there is no binding, there is no cluster generation, and no data produced. So we have to have exactly these sequences for us to get your data. But good news is that the vast majority of kits are Illumina compatible, and this shouldn't be a problem. The sequencing primers, these to the most part should be Illumina compatible as well. There is the ability to change to include custom primers, but that depends on the workflow and the desired data output. And so, as previously stated, this is just the DNA insert. So this is how our library is to be oriented and how it is to be read in the 5' to 3' direction. So let's just talk about a few library types. Some of these up here are the most common that we see at Novogene. We certainly see a lot of WGS and WES. So why am I showing you these? So we're looking at all of these library types. And as this slide depicts, we are looking at DNA libraries originating from DNA. So why is this a concern that we should be scrutinizing these types individually? Is because all of these will produce different segments of information, right? So if we're dealing with amplicon-seq, we could have a high degree of repeats, and we would want to offset that with running this on a lane with something else, a divergent species or a different library type all in itself. Metagenomic studies, these typically tend to be pretty well balanced. So the inclusion of something like phi-X might not be needed. So this is why it's important that we first recognize that there is a difference between samples and library types, and as well within is also important as well. We can have with bisulfide studies, there can be a difference between and across samples that would need individual catering. So now we jumped into some common RNA types. The RNA world is blowing up right now in NGS services. Really the total RNA, so our long non-coding pipeline, and our eukaryotic pipeline are the most commonly used. And so it's important, once again, to evaluate these library types individually and potentially what is going to be loaded on your lane with it. So, for example, the strandedness of a eukaryotic library might not be as suited to be loaded on the same lane as a RIP-seq. So it's important to pay attention to how these are being loaded. If you want data output from different library types, something should be considered before just saying, yes, run these libraries on this lane. One thing I should note is that all of these services that I have on this slide and the slide previous, Novogene does offer. So if you have the needs for the precursor to pre-made libraries, please feel free to reach out. We can help you with that as well. So just what's blowing up in the sequencing world right now is the 10x genomics platforms. So the 10x genomics platforms, it's also very important to understand, one, what these capture kits do, and two, how these can affect other 10x libraries if loaded on the same lanes. So up here, we can see that these are quote unquote regular library types, but they have a bit longer reads. And so what does that mean? It means that the cycle number is to be increased. So as mentioned earlier, the theoretical possible number of cycles that the workflow that is currently being used is 338. So with some of these 10x kits, we have to increase the cycle number for in any given indice, depending on the approach and the kit. So it's important to know that sometimes we cannot run a 10x kit that has 4 sets of I7s and a 10x kit that includes just dual index. So there's a difference in the way that we approach and the sequencing cycles that are accommodated here. All right, so and then this, I just wanted to give a brief explanation here, or sorry, shout outs, I guess, if you will, to the commonly used kits that are on the market. These we do see all the time. NEB is a great one. Yeah, so these are just some common library prep kits. All right, so we have gone through, jumped there, sorry. We've gone through essentially everything up until the point of loading the samples on the machine. And so this is where it's important to understand the differences, to understand your data needs, and to understand the potential need for a different sequencing approach. So we do currently still offer the HiSeq platform, and that is only accommodated in the PE150 workflow. So there are 8 lanes per flow cell, and there are 2 flow cells that can fit on this machine. Theoretical data output per lane is around 110 gigabytes. One thing to note is that this is slowly being phased out by Illumina because they are switching their reagents over. So they'll stop producing this shortly. So Novogene will not be offering this for an extended period of time. Now let's jump into the S4 platform. So this is run on the NovaSeq 6000. The S4 is one of the most, the S4 chip is one of the most robust chips that there are on the market for this sequencer. It allows for 4 lanes per flow cell. There are 2 flow cells per machine, so 8 total lanes per machine. The sequencing approach that is most commonly used with the S4 is the paired in 150, and this is what we currently offer for our S4 chips. The theoretical data output here is 800 gigabytes per lane. So this can be purchased, the S4 through Novogene can be purchased by lane. So if you want 800 gigabytes of data, we can sell it by lane, or we can sell by gigabyte on our partial lane pipeline. So partial lane is a good example of ways to just get your sequencing data past concept. So what I mean by that is it's a great way to try out your constructs. It's a good way to validate your inserts. It's a great way to do low pass sequencing. You're able to purchase data amount by gigabyte all the way up until the 800 gig mark, but at this point, it's more advantageous to buy the lane. I'll get into this further as we proceed. The other option that we do offer is on the Nova 6000 is the SP lane, or SP chip, and that can be accommodated by a couple different sequencing approaches. So mind you, shorter isn't always better. It depends on your library construct. If you need only 50 base pairs, and it might behoove you to get it. If your insertion has a lot of homology within the first couple base pairs, you may want to extend to 150, even though your inserts might not be that long. 250 at sequencing paired in approach does have its advantages and its drawbacks. We'll get into that as we proceed. So these are the options that we do offer. We'll get into a bit more on the prime flow cell, so the SP flow cell. As I alluded to, it's designed for smaller runs, so smaller data production. There are different cycle configurations. So we spoke about how the S4 has a cycle potential of 338, whereas depending on the sequencing approach, so if we are doing SEPE50 or paired N 250, the cycle amounts will increase or decrease accordingly. So it's great to get started with your first toes into next generation sequencing. It's a good platform to get going. Having said that, the more commonly used one nowadays is the S4 chip. And on that, I would recommend partial lane sequencing. So we've gone past everything we need to know about Illumina, everything in quotation marks. And here we have gone over library structure and type. Now we're going to talk about some sample requirements and the way we interpret libraries and the quality that they have here at Novogene. So the way that our workflow progresses is that we shall receive your samples at the way that they're sent to us. So if you are multiplexing or if you are not, so if you're creating a pool of libraries or sending us individual libraries in a tube, we'll perform the following steps on all tubes that are given to us. So we need to find out the volume. We'll run it on the bioanalyzer and the qubit. So we'll find the concentration and if there is modality, if the curves look correct, if it deviates from the trend line. We'll also be looking into the qPCR, performing qPCR. This is very important because we need to know accurate loading concentrations to be able to give you the desired data amount. This is our generalized library QC workflow. And then after that, we will give you a report that tells you how we grade all of the samples that are sent to us. And they're given one of three marks, pass, hold, or fail. And within those marks, we determine how we can guarantee data for you. All right, so here let's get into some sample requirements. So at the top row, we still have our HiSeq platform as it is still being offered, but as mentioned, it will be going away. The S4 and SP chips, we ask that the same concentration is sent across the chips. So we ask that your sample is at least 0.5 nanograms per microliter with around 25 microliters to 50 microliters given this platform. I tend to stick or advise my clients to stick with the more accurate approach of the 2 to 3 nanometer that's nanomolar concentrations. The reason why this range is here, the lower end of the chip is right here, so the bioanalyzer can only go so low. And the upper end, we don't want to overwhelm the chip, so the upper end is also around 30. We need accurate depictions of what your sample is. So this is why we ask for them in this range. As I mentioned earlier, you can purchase data amount by gigabyte for our partial lane service. So this is our partial lane service on the S4. The costs and the data amounts depend on your needs and the library types that you will be sending. You can feel free to reach out and we can provide some more clarification here if needed. But as you reach a certain data amount, so really once you get past 400 Gs, it's more advantageous for you to buy your full lane as opposed to do a partial amount. Just one thing of note, the lowest data amount that we can extend for our partial lane service is 30 gigabytes. All right. So as previously alluded, we already went over the concentration requirements. So please send us anything that is within this range. If you suspect it is a bit above, go ahead and dilute it or just inform us and we can act accordingly. So now we're going to get to one of the most important aspects of your library constructs, and that is the insert itself. The inserts, a couple of things that we need to pay attention to is right off the bat should be size. Size incorporation of your insert can lead to skewed data output to one size or another. It can lead to lessened data output. It can lead to bad quality data output if we do not consider the inserts themselves. So here we have the ideal size ranges for the sequencing approaches. So if we think about it, if we have paired in 150 and we are sequencing from the 5' to 3' direction in 150 base pairs, and then sequencing from the 3' to the 5' direction in 150 base pairs, your ideal range should be somewhere near here, such that you don't overlap in reads and your reads do not go into the adapter regions. If they do that, you do tend to get less data output. Also, so this is the ideal size for the given sequencing approach, but something too that we want to pay attention to is too large of inserts. So too large of inserts can really cause issues with cluster generation. Imagine you're sticking your arm on a table and your elbow is facing up at a 90-degree angle. And that itself is the library. And you flop your arm down and the library reaches over and can potentially interrupt cluster generation or sequence biosynthesis of another library. So it causes physical disruption to libraries near it, which can skew your data output and also give less confidence in the read quality associated with it. So the Fred score can be affected. On the opposite end here, it is also important to not include drastic differences in sizes. So if you were to provide multiple libraries in a tube, and those libraries are composed of, let's say, ideal fragment size, so around 320 base pairs for the paired in 150 approach, yet you give us another library that has a 600 base pair insert. So 600 base pairs means that your library construct in total is around 700 to 800 base pairs, including the adapters and the indices sequences. So this can skew your data output towards the smaller fragment, purely because during cluster generation, these smaller fragments will outcompete the larger ones, thus giving a stronger signal and more potential for reads in the smaller fragments. So we want to make sure that we know how we're approaching our inserts in terms of the platform we'll be using and the designated science output that is needed. So here I just want to go over really quickly a way that these are just trace reads. This is one way that we interpret our library QC results. So these are both examples of non-ideal trace reads for a library. So let's go ahead and start with the one at the top. So at the top, we can see here that this region here is adapter contamination. So adapter contamination ideally should be removed if possible. If not possible, we're only looking at a small fraction, I would say, generally, of a data skew. You may lose a portion of the requested data to this portion here from physical interference and possible non-target incorporation. So we have some sort of adapter primer dimer contamination here. As well, we can see there's a broad peak here, which means that maybe there was greater fragmentation of our library insert. And so imagine if during clustering, we had somehow a shearing of our insert, it could have produced a different fraction of the library that is off target or is not of interest. So having said that, it's important to take this into account. Whether or not you'll get good data here, I would venture to say yes, but it's always best to optimize as best we can. Moving down here, this one I would say is more of a concern depending on your library type. So multiple modality is not common for target genes or for targeted regions. For things like ATAC-seq, we do tend to see multiple peaks, and that is normal. But here, what we're seeing is we have some contamination. So this, once again, is primer and adapter contamination. Here, we are seeing 2 peaks. And what this implies is that a vast majority of your sample has an insert with around 300. But you also have potentially a secondary insert. Maybe it's off target, maybe it's on target, we don't know. But we do know that if you sequence as is, the likelihood that you'll get the same data representation here as you will here is drastically changed. So it's important to know that we most of the time don't want to do this. And if we can separate the libraries by size, that's the best approach to go. So quickly going through just some common ways to get rid of these abnormalities in your library. So if we have adapter primer contamination, just go ahead and do a bead pull down. That should remove any non-target things that are still in your prep. Small insert size. So this is an issue if you're, depending on the sequencing approach, if your insert is 60 base pairs, but you're sequencing 150 paired in reads, you're going to get into your adapter sequences and you'll basically just giving up data amount there. So it's best to redo the prep and either a different approach, sequencing approach, or shorten your insert. Broad peaks in modality, as mentioned past, multiple modality, data output is skewed, lower data. So go ahead and do a bead pull down or a gel isolation to separate your fractions. That should resolve that there. Concentration, so it is important that we have at least 2 nanomolar of your library. This will allow us to provide the quoted data amount. We can proceed at any risk that you're comfortable with, but generally, the less physical amount that you put on the lane, the less data that is produced. So these are just typical ways to amend any potential errors that you might get in your data quality control or your library quality control. So some quick things. So for us, we have a platform called CSS, and this is the ability for you as the client to upload all of your sample details in there to your own individual portal. This is where we would request that you would put that. Now, given that our QC requirements are around, for example, say 0.5 nanograms per microliter, we do tend to ask that you send more. This benchmark that I gave previously is for our post-QC samples, meaning that, yes, it meets our qualifications, but there may be potential for human error here. You might have a run stop. The power might go out. So we'll need more of your material to get the quoted data. One thing that is really important to note, we do not recommend that you pool more than 50 sublibraries into one tube. This is for a couple reasons. It is hard, near impossible for Novogene to guarantee individual sublibrary data distribution. It is also hard for us to guarantee that it will be equal in quality and distribution across sublibraries. In short, we generally recommend that you send us all of your individual libraries, allow Novogene to do the pooling for you. This removes the responsibility on your end and puts it on Novogene, and we have a lot of expertise in doing this. So I think we'll be better suited if possible. All right, so now we're just gonna get down to sequencing and demultiplexing. We're running a bit short on time here, so I'll see if I can get through everything. So let's just refresh ourselves with the generalized library construct, right? So we have our DNA insert in the center, flanked by our two reading primers, so read 1 and read 2. On the outsides of those, we have our indices, indices sequences, and then the oligos, which is where the construct itself binds to the flow cell. So generally, for the most part, across all of our platforms, we do request that common Illumina read sequencing primers are used. And these are the ones that are generally used here. There are some other accommodations that the flow cells and the lanes make, but they're not as common. If you have needs for special primers, if you are using a non-kit approach to construct your libraries, if you have the greater need for demultiplexing and you've included some UMIs or inline barcodes and you would like to change the diversity of those, sometimes clients will provide custom primers. So one thing just to point out, the high-seq lane, we can only accommodate a change in a custom primer for the I7 only per lane. The I5 cannot be changed. And the reason for this is that, as mentioned earlier, there are 8 lanes on the high-seq flow cell. Apologies. 8 lanes on the high-seq flow cell. And each individual lane can have its own potentially customizable I7 primer introduced. The I5 is introduced across the flow cell, so only one optimization here per lane. If you purchase per flow cell, we can accommodate almost any request, given that the indices, their lengths are limited to 8 or 6 base pairs, and that is, again, a limitation on the high-seq workflow itself. We can accommodate other primer and indice changes, but for the high-seq, but that is also only on flow cell basis. We can do pretty much any other arrangements, given some more validation by our backend teams on the S4, but that is also, once again, by flow cell, because of the same reason that was alluded to earlier with the I5 indice or the I5 orientation on the high-seq. So this is just a general idea of what custom primers we're able to accept and any accommodations that we can make. For our partial lane service, so if you're to order less than a total lane, so let's say 50 gigabytes of data, we require that you, 1, provide us with all indices, both I5 and I7, if applicable. Sorry, if the I5 is applicable. And no customization is to be done. And this is because we will be loading your samples on the lane with potentially in-house libraries and other clients' libraries to share the cost of the lane. We need to be able to distribute primers evenly and effectively across all samples on those lanes. And we cannot do that with customized sequences for our partial lane service only. Let's get into one of the other final steps that we're coming up to. So we've gone over the library constructs, we've gone over the machines, we've gone over the sequencing approaches, we've gone over how to insert and what to insert into your constructs. Now, let's go over what might be what might need to be added to your run. So one of the most commonly sequenced things in the entire world happens to be phi-X. It is a phage, and that is one of the earliest things that was sequenced. It's well known, it's conserved, it's diverse, has a very small genome. So what is done in a lot of NGS sequencing this day and age is the addition of the phi-X genome is added to a sequencing run. And what this does is it will offset some potential issues that may arise given a library type. So let's go over low diversity libraries. So why should we add phi-X to low diversity libraries? Well, if we have, let's say we purchase a lane and we have included 10 libraries on this lane, and we want it to be sequenced and we want even distribution. Well, when we have a low diversity library, it's hard for the machine itself, the sequencer itself, to decipher the base calls. Imagine if you have a stretch of A's or T's and all of the outside signal noise, so everything else on the chip is flagging A. But in your true sequence, there is a base that's a G. Given the outside noise and the calls previous, the machine might not be able to detect a varying base. And this is purely because of the resolution of the signal to strength ratio. So we can offset that by including known sequences to validate a potential base call. So this is why we would want to do that for low diversity libraries. And in some cases, it is a big proportion of the lane. So for GBS libraries, we would recommend near 20% of the lane be associated with phi-X to overcome this issue. Now, it is important to note that whatever percentage you give up to phi-X, you will be removing from the data output. So you have to be comfortable with the potential loss. Having said that, the loss in most cases will outweigh the potential degraded base calls. So unbalanced libraries, very similar. If we have say at the start of your sequences, you have inline barcodes or you have some UMIs, and those are constant across all of your libraries, they're all the same. We want to make sure that we introduce some variability there. So the machine, the sequencer itself, doesn't halt the run and say, hey, something is wrong. I keep getting the same reads. Is it true or is it an artifact? So we add phi-X to offset that. And another thing, potential variable here is fixed bases, and it's very similar to unbalanced. If you have fixed bases in the start, the middle, or the end of your insert sequence, we potentially need to accommodate that with a greater or lesser need of phi-X. For our partial lane service, phi-X is actually normally not a concern, right? Because we are loading your samples on a lane with other library types and other species. So the diversity and the balancedness of the libraries should be just great the way that they are. But if our lab does need to make accommodations, we will add phi-X, but that does not eat away at your total data output, given the qualities of your libraries after data QC, library QC. So once again, let's just get through some quick information here on indice orientation. So as alluded to earlier, the way that your library should be constructed is 5' to 3' orientation. That is read as the arrows depict here. So for us, we ask that you return the forward workflow. Even though earlier I mentioned that we sequence using the 1.5 reagent kit for the Nova's platform, our demultiplexing software actually uses the V1 or the forward orientation. So this is the orientation that we ask that you provide your indices in. We will be sequencing in the reverse orientation, but our demultiplexing software will use the forward orientation. And so it's also recommended that we try to avoid more than 10 libraries to be pooled per lane. So you can send us more than 10 libraries to be loaded on a lane, but try to avoid sending us more than one tube with 10 sub-libraries in it. It allows for us to do further demultiplexing separation and to control for the library data output. So real quick, I just want to go over the potential index changes that are possible across platforms. So for the HiSeq, the only orientation possible, the only length possible is 6 base pairs and 8 base pairs. No other accommodation can be fit for the HiSeq platform. For Novogene S4, we can essentially change the index read lengths to almost any amount further evaluation might be needed depending on your specific needs. Our only limitation really here is the 330 cycles. Same limitation will apply to the partial lane. We can do it to an extent. Whether or not you want greater than 36 base pairs, we cannot offer that on our partial lane service. It has to go through full flow cell. So the SP flow cell has a little bit less accommodating ability. So here with just the single end 50, that is just reading the I7. And then with the PE 250, we can accommodate the I7 or I5 at 6 or 8 base pair. If you want to do 10, full flow cell is needed because we have to spread that out across the flow cell. Okay, really quick, this is just gets back to why it's important for us to understand our index approach, our insert approach, is because of some different library structures, right? So here is the 10x example. These are the read lengths and the indice lengths. So for these common ones here, we don't have an I5 index. And so this can be run on potentially any platform that we have. The single cell ATAC-seq has a different approach where they have 8 base pairs and 4, they have an indice of 16. So this approach may change the platform that you can operate on. So here we can see that this cannot be done on the HiSeq. And there's some more different variations here as we progress through the 10x platforms, the multi-ohm targeting kits, the gene expression kit. Flow cells most of the time are required. And then let's go ahead and wrap this up with data quality control. So we have figured out how to get everything on the machine, the best orientation possible, the best sequencing approach for our given set. Now let's try to talk about data quality control. So for us, what will be done is that every single one of your reads will be filtered to remove any adapters. So what I mean by this is in read adapters, we can give you both raw data, and if requested, we can give you trimmed data. So we can trim to any length. We can also remove any inline barcode reads. And so that is if your insert is shorter than the 150 bases of the read length. We'll remove any low quality reads. So if the read score is, if the read Fred score is too great and 10% or more of the reads are like that, we remove that, and if needed, sequence at further depths. Also, we'll remove low quality Fred scores. So this will also give you, the report will also tell you the GC distribution, data output error rate. This is our base platform, or sorry, base data quality control, and it's given to every client for free. So we've gone over what is needed to get your sequences in a construct, from a construct to the machine, from the machine to the data production, and then here, the data analysis. And with that, I would just like to thank you for your time. And as Chloe mentioned at the start of our session, if you do have questions, please go ahead and drop those in the chat box, and we can get to that as soon as possible. Thanks everybody for their time.